I'm Bob Handsaker, and I work here at the Broad Institute and at Harvard Medical School in Steve McCarroll's lab. And I'm going to be talking today about Genome STRiP, which is software that we developed for doing structural variant detection in whole genome sequencing data. Genome STRiP is for whole genome data. After I talk, Menachem Fromer is going to speak about XHMM, which is a tool for doing copy number variation detection in exome captured data. So Genome STRiP's whole genome, XHMM is for exomes. So the rough outline of what I'm going to talk about today, I'm going to give you an overview of structural variation calling in general. I'm going to talk about details of how the processing pipeline works in Genome STRiP, some techniques you can use for quality control if you're doing a large discovery run or genotyping run. I'm going to talk a little bit about a usage scenario where you just want to genotype a small number of sites in a public data set, like 1,000 genomes, and how to get the software and how we provide support and things like that. So in terms of the general overview of structural variant calling, the problem is that we want to ascertain these large structural variants. And in terms of definitions, I'll say structural variants are effectively something that's longer than a read length or longer than a couple of reads, right? If you're smaller than a read, you can use tools like the GATK and you can find small indels. But if you want to find something longer than a read, we have this problem. We have little short reads and we want to get really large variants. And there's 3 or 4 sort of themes that people have used in structural variant detection software. One is to look at, if you have paired-end data, to look at the read pairs, because you have an expectation about how far apart the read pairs should be. You can look at if when you map those read pairs back to the genome, you get a different spacing than you would expect. And you can use that to detect structural variations. You can use that signal in the data. The other thing you can do is you can just look at read depth. You just count how many reads land on a certain portion of the genome, and you look to see whether there's more reads than you would expect or less reads. There's noise and variability in the read depth, and it varies with GC, but you can use this signal also to help detect structural variation. You can also look at split reads, especially if you have longer reads today, 100 base pair reads or something. If there's a breakpoint, like a deletion in the genome here, you may be able to find reads that actually cross the deletion breakpoint, and by looking at both ends of the reads, you can detect the deletion and do it with base pair resolution. And the final sort of general theme is that you can just do de novo assembly and then map all the reads back to the genome. And there's pluses and minuses of doing this. Assembly is a difficult problem. And Genome STRiP really tries to integrate these first 3 methods. We don't do de novo assembly, but we try to integrate these 3 different signals from the data in different ways to do structural variation, discovery, and genotyping. So if we have all this sequencing data, right, what makes structural variation calling challenging? Because if you look at results from different callers out there, you'll see that sometimes there's a high false discovery rate and people are confused about why it gets difficult. And one reason is that there's a lot of artifacts in the sequencing data. The sequencing data isn't perfect. And we get these artifacts which can mimic the same signals we're looking for to detect structural variations. So for example, just the laboratory preparation techniques result in these chimeric molecules, right? So this is a plot down here of a section of all of chromosome 5, actually. And it's looking at all the aberrantly spaced read pairs. And you can see it's pretty uniformly distributed over most of the chromosome, except for kind of a gap caused by the centromere. And these are all chimeric molecules that are created. So the presence of a single molecule that's mapping to places on a chromosome or even across chromosomes isn't necessarily a guarantee that there's a structural variation there. It can just be an artifact like this. Read depth has sources of noise, as I mentioned. Read depth, if you look at read depth from whole genome sequencing, it will tend to have a lot of variability, even more in exome capture, because the capture step adds noise to the read depth. And repetitive structures in the genome can be misled, can cause misalignments, right? Especially in the presence of sequencing error. And so this adds additional complications that you may not know for sure that a read maps to where you think it does in the genome. Low coverage sequencing makes this problem even more difficult, although in our experience, while depth helps, right? Methodology is actually more important. If your methodology isn't rock solid, when you add more depth, sometimes callers get more confident in their mistakes rather than getting better. So Genome STRiP, the idea behind Genome STRiP is that we want to combine both the technical features of the data, these split reads that I talked about, and read pair spacing, and looking at the depth signal, and we want to combine those with population scale patterns that we expect in humans, right? Because most structural variation that we're going to detect in normal people, not looking at tumor normal cancer projects, most of what we're going to detect is actually shared variation. It's shared between different members of the population, and so we can aggregate information across people to get more power to detect variation. Also, because the population is heterogeneous, we can look at the distribution of the evidence across people and look to see if we really are able to categorize people into being homozygous for the reference or homozygous for carrying a structural variant or heterozygous. And then we can look at the haplotype structure in the human genome to get further information about exactly who's carrying which variant. So Genome STRiP is this toolkit of all these methods for discovering structural variation. In particular, I'm going to talk about our pipeline for discovering and genotyping large deletions. So in the 1,000 Genomes project, which is really where Genome STRiP was developed, we've had very good results in applying Genome STRiP to this 1,000 genomes data. 1,000 genomes is low coverage 4x sequencing. And we've consistently posted a very low false discovery rate based on validation experiments done in 1,000 genomes, usually in the 2 to 5% range. I don't think we've ever been above 5%. Our sensitivity has been quite good. We're not perfect, but in phase 1 of 1,000 genomes, about 80% of the deletion call set was called by Genome STRiP. A lot of variants were called by multiple algorithms, but if you did the Venn diagram, 80% of the total was called by Genome STRiP. And Genome STRiP was used to do all of the genotyping for large deletions in both the pilot and in phase 1 of 1,000 genomes. And our genotyping accuracy overall, benchmarked against this Conrad array-based data, was 99.82% in terms of genotype accuracy. Genome STRiP has also been used in several other large projects. In addition to 1,000 genomes, the Genome of the Netherlands project, which is wrapping up, which was 750 samples in a trio design. The GoT2D, type 2 diabetes consortium, the UK 10K project, which was at 6x coverage. And right now we're calling 1,000 genomes phase 3, which is 2,500 individuals. So these are some of the large projects Genome STRiP has been used in. So I'm going to talk a little bit about the processing pipeline. There's a lot of code snippets in here, which I'm not going to dwell on in the interest of time, but I want them to be in the presentation because this presentation also gets used as a sort of a resource later people can look at and look at examples and example pipelines and pieces of code. So at a high level, in Genome STRiP, one thing we do is we separate the process of discovery from genotyping. So discovery in Genome STRiP is just figuring out whether or not there's a site that's polymorphic in the genome. And then genotyping is trying to assign to each sample their exact genotype at that site, whether they're homozygous for a deletion, heterozygous, or whether they're homozygous for the reference. And by splitting these two things up in the software, it allows us to sort of combine things in different ways, right? You can go in with a new data set and you can discover novel polymorphic sites, or you can just genotype known events in new data sets or in existing data sets. And this has also allowed us to take calls made by other SV callers in 1,000 genomes, for example, and then genotype them in Genome STRiP. And that's the way the 1,000 genomes project deletion calling was done. So if you drill down one level and look at sort of the next level of detail of the pipeline, there's this pre-processing phase where we take the BAM files and we pre-compute a lot of information that's used by later phases in the pipeline. there's this discovery pipeline you can run, and what you get out from discovery is a VCF file that has the polymorphic sites, but no genotypes for each sample. Genome STRiP doesn't have a module to do breakpoint assembly, but if you want to use split reads in genotyping, you need to have base pair resolution breakpoints. So if you run a separate breakpoint assembler, then you can feed that into the genotyping pipeline, or you can genotype without having found exact breakpoints, in which case we'll just use read depth and read pairs to do the genotyping. And then so we have a sort of separate track for doing genotyping that can come from sites found by Genome STRiP or not. You still have to have the pre-processing data, but the VCF can come from anywhere. The input here to genotyping would be another VCF. And then what you would get out is a VCF decorated with all the samples and their genotypes. And then you can optionally, I'll talk just a little bit about doing genotype refinement by using LD structure in the human genome. So to run Genome STRiP, these are the 6 inputs you need, right? You need analysis-ready BAM files, which you probably heard about this morning, what this means in terms of the GATK. You need your reference sequence, an index FASTA file. You need something called an alignability mask that the Genome STRiP uses to determine which parts of the reference genome can be accurately aligned and where the aligner is going to have trouble. This has to match the reference you're using, and on our FTP site, there are alignability masks available for download. There's also a copy number 2 mask where this is picking out regions of the genome that we believe are not variant in most people, not variant in copy number, and this is used as part of our GC bias correction. There's a ploidy map and a gender map that I'll talk about in a little bit, and I'm going to talk about each of these in a little bit more detail, so I'll just go on. So the analysis-ready BAMs, as I mentioned, you probably heard a lot about this morning. It's essential that you do marked duplicates. Indel realignment really doesn't matter. If you've indel realigned your BAM files to use them with the GATK, it's fine. You can run them through Genome STRiP, or you can run them without. As far as I know, it doesn't really make any difference. It is critical that Genome STRiP, almost all BAM files have this, but it's critical that you have these key tags, right? You have to have the read group identified. You have to identify the libraries, the samples, and the platform that the reads came from, whether it's Illumina, for example, or some other platform. We don't support GATK reduced reads compression today, so if you have a reduced BAM, you need the original BAM to run Genome STRiP. As I mentioned, these alignability masks help us understand whether or not we should trust the mapping at each position in the genome. And we do this by computing across the reference how unique each position in the reference is. And in particular, we're taking all k-mers across the reference and just asking whether or not they align uniquely back to the genome. And you need to pick a value of k to use with your alignability mask, and you should base this on your read length. If you have 100 base pair reads, use 100 for k. If you have a mixture of 50 and 100 base pair reads, the right thing to do is use 50 for the smallest reads you have. Or you could try calling in batches and use 50 for your 50 base pair data and 100 for your 100, if you wanted to do that. There are a bunch of pre-computed masks that you can get from our FTP site for common genomes and different values of k, or you can build your own. If you look at the documentation for compute genome mask, and you can just feed in your reference and what value you want for k and build your own mask. Whoops. So the copy number 2 mask that we call it is used in GC bias normalization. So if we're going to look at GC bias across the genome, we don't want to get noise from common copy number variants in the genome. So we use a method that was originally pioneered by Peter Sudmont, where we start with this mask of regions in the genome where we've excluded all the regions from DGV, we exclude the sex chromosomes, anything where there's a lot of repeats from repeat masker, and then we use this relatively clean section of the genome to estimate GC bias. You can also download the copy number 2 mask from our FTP site. I guess there's no officially supported tools to build your own, but if you need to, you can write to me. If you want to make a bed file, for example, for another organism, you can convert a bed file to a mask like this, and somebody was doing that recently for another organism. The next input you need is this Ploidy Map File, which is simply a very simple text file that tells us what the expected ploidy is by gender on the genome. And for common reference sequences, you can download one from our FTP site again. And it's simply, in this case, marking out where the pseudo-autosomal regions are and where the sex chromosomes are. So we know what the expected ploidy is. So in the copy number 2 mask, we exclude the sex chromosomes because we're trying to get a global estimate of GC bias in each library. So what you can, and there's also a tool in Genome STRiP if you want to plot like the GC bias curve. There's also a tool in Picard to do this. But in whole genome sequencing, the efficiency of the library preparation and the sequencing varies a lot with GC. So if you ask whether or not the coverage is even across the genome, and you look at the genome broken up into bins of different GC content, you'll see that in the middle range of GC, you'll have very good coverage, and as you get to very high and very low GC regions, you'll get a drop off in coverage, right? So when we're analyzing read depth, we correct for that GC bias. But to figure out how much to correct, we just look at a very clean, a large, usually about 500 megabases, but clean region of the genome to get a very accurate GC bias profile. Does that make sense? And then the ploidy map is used in a sort of a different context, right? When we go to call deletions, for example, on chromosome X, we want to call the pseudo-autosomal regions different from the other part of X, and we want to call it in a gender-specific way, right? So the ploidy map is used to control for gender and to allow you to call in the sex chromosomes. You actually don't need the ploidy map if you're not going to call in the sex chromosomes. If you're just going to call in the autosome, you don't need to supply it. And finally, in conjunction with the ploidy map, we need to know the gender of each sample. Genome STRiP doesn't try to estimate gender internally, so you have to supply a file with the gender of your samples, again, if you want to call on sex chromosomes. Is there a question? So the other input that goes into Genome STRiP is the configuration file. Usually you don't need to modify it. In older versions of Genome STRiP, you had to modify the configuration file to put in information about your reference genome, but this isn't required anymore in modern versions. And also, all the tools in Genome STRiP now will allow you to override the configuration parameters using -P. So you don't have to go in if you want slightly different settings for some reason. You don't have to go in and actually edit the configuration files. So those are the inputs. And in terms of the steps in the pipeline, I'm going to sort of walk through each step, and I feel like I'm getting a little behind in time, so I probably need to pick up the pace here a little bit. But a lot of this is just code that's mostly there for reference. So the preprocessing step is actually pretty important, and it is, you know, a substantial fraction, maybe a third of the cost of running Genome STRiP in general. And we compute a lot of information about the data, right? We look at the insert size distributions in each library. We look at the sequencing depth. We're estimating this GC bias. We're looking at not just the insert size distribution itself, but also if you sort of integrate that over the genome, how it looks. So there is this preprocessing step that gets run, and this metadata directory gets created that has a bunch of files in it. So if you look in the metadata directory, you'll see that we're calculating sort of this effective genome size based on the mask you used. We're calculating the insert size distribution data in a couple of different formats. If you have a large data set that you're processing, it's important to use this reduce insert size distributions flag, which causes us to make a lossy compressed version of the distribution so we can use them at runtime more efficiently. It isn't actually documented here, but there's also a tool that allows you to plot and look at the GC bias profile, so you can see what the GC bias looks like in your samples. Actually, I think, to be honest, I think there is a slide on this a little bit later. So all the main pipelines in Genome STRiP also use the GATK I don't know what you call it, facility for running multiple jobs and parallelizing your jobs and running your jobs on a cluster. So everything in Genome STRiP is designed to be parallelized, so you can do big computations and things run through these Q scripts. So when you're running preprocessing, you have to say the output metadata directory where we want to store all the preprocessing information. You put in the masks. And very useful flag is that if you don't have any BAM files where the same sample is in more than one BAM file, you can tell Genome STRiP that the BAM files are disjoint, and this speeds up processing because we don't have to worry about a sample being split across multiple BAM files. And of course, you put in your BAM files, and then you run preprocessing. There's also a new option in fairly recent releases of Genome STRiP. It used to be that Genome STRiP made 5 passes over the BAM file. It would do sort of each of these preprocessing things in one pass. And so now the default is that it will do it in 2 passes. It does 1 pass to get a good look at the insert sizes, and then a second pass that does everything else. Generally, 2 pass is better, but if you have a lot of CPU available, sometimes multipass can be faster. And in some benchmarks we've done on the Amazon cloud, maybe multipass is faster in some configurations. But 2 passes is the default, but there's a flag so that you can override this if you want to go back to doing it in multiple passes, if that's faster in your pipeline. Another new feature we've added recently is that you can now use multiple metadata directories. So when you do preprocessing, each preprocessing run will create a single metadata directory. But then if you wanted, for example, preprocess your samples in batches and then combine all those batches for discovery and genotyping, you can simply pass multiple metadata directories in. You don't have to take 1,000 samples and 1,000 samples and then put them all together and rerun preprocessing on the 2,000 together. So once you've done preprocessing, one thing you could do is move on to doing discovery in your data set. The idea behind discovery is we look at aberrantly spaced read pairs and we look at read depth across the genome. We don't actually use split reads in discovery, and we're looking for sites where there's signals of deletions, looking both across your samples and within each sample. So the inputs, again, are the metadata that you've pre-computed, your BAM files, and what you're going to get out is a VCF file. It's important to know that the output VCF file contains every site that was evaluated by Genome STRiP. Most of those sites are, in fact, not polymorphic, so it's important that you look at the filter field, because only the ones that say pass are the ones that we've decided are polymorphic. Partly for debugging reasons, we generate outputs on every site. This also allows you to go back and change the filtering. If you're an advanced user and you think you're missing some good sites and you want to adjust the filtering a little bit. If you look in the Q script, you can see the default filters that get applied. In terms of how, if you're integrating this into a pipeline, we do it in parallel per genome locus and also per length range. So you can look for small deletions in one run and large deletions in another. And this is a very useful thing to do in the current implementation because there's some optimizations to make the performance better on sites that are 100 KB or less. So if you want to call things that are greater than 100 KB, it's useful to do that in a separate run from the run where you call things that are less than 100 KB. So this, again, is just for your reference, a typical invocation of the Q script. Here's where we're parallelizing based on event size. We're saying we want deletions ranging from 100 bases to 100,000. The run directory is where a lot of intermediate files get produced, and these are used downstream for QC and reporting. And what you get out is this VCF. Also, when you parallelize, these arguments here tell you how much of the genome you want to run. In this case, we're saying to run a megabase at a time with a 10 KB overlap. This window size needs to be larger than the maximum size you want you want to discover. And the overlap, this 10 KB overlap, it really just has to be bigger than the, a typical insert size. So, but, going from 10 KB to like 1 KB doesn't speed things up significantly. So you can be very generous in giving a large window padding. If you look at the output from discovery, you'll see that in addition to the samples that passed discovery, you'll see a lot of sites that were filtered out due to these various filters. And these are what the filters mean, right? If you want to interpret the VCF file that comes out. So the coverage filter just says that the site had either excessive read pileup or there just wasn't enough coverage to make a good call at this site. The coherence filter says that the read pair spacing was not consistent with there being a single segregating deletion at that site. It's remotely possible that there were 2 very close deletions, and sometimes sites like that can fail the coherence filter, but in general, the coherence filter is pretty good at picking out good deletion sites. The depth and depth p-value filters are asking whether or not the read depth and read pair signals look consistent, and whether or not there was enough depth to get a good measurement at this site. And pairs per sample is a filter that by default is essentially requiring that we see at least 2 read pairs in some sample before we will believe that a site is real. And this filter is very good for improving the specificity of the calls. And you get these filters if you just run out of the box. One filter that doesn't just run out of the box because you need to also supply a UCSC track is an alpha satellite filter. Generally, in regions of alpha satellite repeat near the centromeres, you will get a lot, not a lot, but you'll get a few percent of deletion calls from Genome STRiP. And basically, certainly Genome STRiP can't call anything accurately in those alpha satellite regions. There's a lot of mismappings of the reads that happen in the alpha satellite. I think because they aren't completely represented on the reference, a lot of them are in the centromere, and it's very repetitive with a lot of small difference. And so aligners will tend to align the reads all over those, and there's a lot of signal of structural variation. So we just actually filter out anything with more than 90% alpha satellite in it is typically what we do. So this is just telling you how you can run the alpha satellite filter. So breakpoint assembly, as I mentioned, is not something that's directly supported in Genome STRiP. You have to use some third-party tool if you want to do this. For example, in 1,000 genomes, we use Tegra SV, but you could also use something like Velvet, or you could consult a catalog of known breakpoints. For example, you could use the breakpoints that were found in 1,000 genomes. In general, Genome STRiP gets very close to getting the breakpoint right. Typically, we're within 10 to 20 base pairs of the actual location of the breakpoint. This is a plot showing Genome STRiP's estimate of where the breakpoint was compared to the actual breakpoint when a breakpoint accurate sequence could be determined. This peak right here is actually caused by target site duplications in Alleles, and this was from, I believe, I believe this was the early phase of the 1,000 genomes. Yeah, in 1,000 genomes we used Tegra SV, and we also used Age from Yale to assemble breakpoints. We actually both were run, and then we took a consensus of the breakpoints between the two. If you do have base pair level resolution in some of your calls and you want to use split reads, this will be done automatically by Genome STRiP. If you feed in a variant with base pair level resolution, unless you disable it, we'll automatically use split reads we'll look for reads that indicate the deletion allele rather than the reference allele. There is a module in Genome STRiP. It's mostly historical at this point, and it does alternate allele alignment. And what this is used for is only to get completely unmapped reads. So for example, in the sort of de facto standard in BAM files is if you have one end of the read pair mapped and the other end doesn't align, it's still stored in the BAM file at the location of its mapped mate. If that's the case in your BAM files, Genome STRiP will pick up that read and it will try to do a split read analysis on it anyway. The only reads it won't use is if the pair is completely unmapped, both ends, right? And if you want to actually use those reads, you can run this alternate allele aligner that will go through all the unmapped reads, it'll go through all the junctions in your VCF, and it will try to find completely unmapped reads that may support that alternate allele junction. This was more important for short reads, 36 base pair, 50 base pair reads. With 70 and 100 base pair reads, there's almost no advantage to doing this alternate allele aligner. So it's still in the toolkit, but it's really not that useful anymore because the technology's evolved. And this is the QStrip if you wanted to run it. So whether you got your site list from Genome STRiP or from another tool, if you want to use genotyping, this is how you would do it. So Genome STRiP genotyping, like the other parts of Genome STRiP, uses multiple signals from the data to try to estimate the total copy number and the state in each sample. So we'll use read depth, we'll use pairing information, we'll look at split reads that split across the locus. This plot is showing sort of how the likelihoods improve as we add each kind of data to get a full picture of the locus. And then we will look at this on a population basis to separate, in this case, the reference samples here in blue from the heterozygous samples in orange and the homozygous deleted samples in green. For genotyping, again, the inputs are the BAM files, a site file, and then the pre-computed metadata. And what you get out is a Genotype VCF file. And it's important both to look at the filter field again to see whether or not this site passed genotyping filters, whether or not we think the genotypes are good. The non-passing sites will still contain genotype calls, but you shouldn't trust them. You should consider them all no calls if the site itself doesn't pass. And also each genotype will be marked as to whether it passes quality thresholds or not. But in general, you can use those genotypes, but take into account their quality, but don't use the site at all if it doesn't pass the site level filters. And the workflow here is parallel per site from the VCF, so you can essentially split your VCF up into multiple little sections and run it. Those parallelization arguments are down here. If you say parallel records 100, it'll take each 100 sites from your VCF and run those in parallel and then merge the results back in the end. So when you get a VCF out of Genome STRiP, it'll have the genotypes for each sample. So it'll sort of look like this, right? This is a passing site. And then the format tells you that it's got these, the GT field, which is the genotype, it's got a filter field, and it's got likelihood and quality fields here. So this is standard VCF format if you've seen this before, and if you were at the GATK, you've probably been seeing a lot of this. So the genotype here is telling you whether it's referent or alternate, so this would be a heterozygous deletion. We do, by default, 2 filters on each genotype. We'll tell you whether it passes or whether it's low quality. Low quality, the low quality filter is just whether or not the genotype quality is below 13. So anything that's passing is a 95% confident call. As long as the site passes, you can use sites with GQ below 13, but you just have to realize that the error rate is going to be much higher, right? It's going to be calibrated to GQ. We also make genotype likelihoods, and we do those in 2 forms. We do a genotype likelihood that takes population frequency information into account. So this is our best likelihood estimate at the site. We also generate a GL0 likelihood, which doesn't use any frequency prior. So if you're going to do imputation on top, a likelihood-based imputation, you should use the GL0 likelihoods instead of the GLs, because the imputation engine is presumably going to take population frequency into account. And we emit the standard GQ field that gives you the Fred-scaled quality of the genotype call at that site. There are a number of filters we apply that we generally apply to genotype output. If you just run Genome STRiP out of the box, not all of these filters are applied, and that's because some of them are incompatible with using breakpoints. Some of the depth-based filters need to sort of be applied internally only to the depth signal, and we don't do that automatically today. So you need to do the filtering a little bit differently depending on whether you're using breakpoints in your genotyping or not. So I would say our current best practice, certainly what we did in 1,000 genomes and several of the other large projects, is we ended up not using the breakpoints, in particular because we found that there was a certain level of error in the breakpoint assembly. And so we wanted to not have that error in the breakpoint assemblies influence the genotyping. If the declared breakpoint is wrong, you will actually get pretty inconsistent results for the genotypes. So if you call a site, but you have the breakpoint off by a few base pairs, using split reads across that breakpoint will give you very inconsistent likelihoods from the depth signal or the pair signal. So if you're not using split reads in the genotyping, you can use the script that's mentioned on the next slide, which automates this set of 6 filters. And you can see what we're filtering for here. We're looking really to see if there's sufficient alignable bases, whether or not we got good cluster separation in our depth-based genotyping. We look at population-based metrics like inbreeding coefficient. Genome STRiP also does a couple things that are pretty novel. We'll look at the genotype likelihoods, and if a site looks non-variant, if it looks like all your samples are reliably copy number 2, we'll tell you that site just looks non-variant, you should filter it. And finally, if you give Genome STRiP two sites that overlap, Genome STRiP will look at the posterior likelihoods and try to decide whether these were duplicate calls of the same event. And this was used a lot in 1,000 genomes where we were integrating calls from multiple callers. And we used the posterior likelihoods in the genotyping in Genome STRiP in phase 1 of 1,000 genomes to try to get rid of duplicate calls that were made by different callers but with slightly different boundaries. So if you wanted to run these manually, these QC steps all use this annotation framework that's in Genome STRiP. And in this annotation framework, you pass in your VCF file and you say which annotations you want to run. And this is the list of annotators we run by default. And then you will get a bunch of reports in this output report directory. And then you can and I should have said that in addition to the output reports in the directory, which are text files and easy to go through and process yourself, you also get an output VCF file where a lot of this information has been written into the info field. So you can use that info field then with the standard GATK tools to do filtering. And these are kind of our best practices filters that were used in 1,000 genomes. But in the very latest release of Genome STRiP, we also put out this script, SV Genotyper without split reads, that automates this whole process. But you should only use this if you don't want to use split reads in your genotyping. And finally, just a note on genotype refinement. If you look at what was done in 1,000 genomes, we used a combination of Beagle and Mac to do genotype refinement. So after the genotype likelihoods were produced, we combined those with the likelihoods for SNPs and small indels to create phased haplotypes. And one side effect of doing that is we get much better, not much better, we get improved, because we already have like about 98% complete genotyping of the deletions from Genome STRiP anyway. But we fill in that last 2% with calls based both on the likelihoods computed by Genome STRiP and likelihoods from LD with nearby SNPs and indels. And so we get two things. We get a little bit better genotype calls, and we get phased haplotypes. And this final step of processing, which is more than I can really go into here, was done mostly at the University of Michigan in the 1,000 Genomes project. So I want to talk a little bit about quality control and sort of QC methods that you can use, because one of the problems is you run all this software, right, and you don't know whether you should trust the results. So there's a couple tools you can use in Genome STRiP to help get a handle on quality. And some of the qualities built in terms of our filters are getting more robust, and there's more filters built into the standard pipeline. So it's getting, the situation's better than it was, 6 months or a year ago in terms of being able to trust the data that comes out as long as you look at the filter field. But there are some other tools you can use. So one thing is it's pretty important in Genome STRiP that you have well-behaved insert size distributions. And so there's this tool, plot insert size distributions, that you can use, and it will show you the insert size distribution for each library. So here's a typical plot. And if you look at the output in Genome STRiP, you'll see that it actually makes 2 plots for you, right? And the one on the left is the one that you'll see from many different plotting tools that are plotting insert size distributions. So you see that there's a big peak, and it's at around 500 base pairs, maybe just where you think it should be, and the standard deviation looks good. But Genome STRiP also makes the secondary plot where it blows it up by a factor of 10. And in particular, we do this so that you can see the right tail. Genome STRiP is very, very sensitive to any excess mass in the right tail. This one is pretty clean. There's a little bump here and a little bump here. But if you're seeing any significant mass in the right tail, then you maybe want to not trust the calls from that library so much and maybe exclude it from analysis. Either that, or there actually is a way you can override the estimation of where you should start to consider the read pairs as being aberrant library by library. And so if there's a library that's got some extra mass out here in the right tail, you can actually still process that library, but at a different threshold. In terms of understanding the output from discovery, this is an example from the GoNL project. And this is mostly just to emphasize that in GoNL, from the 250 trios, if you look at the discovery VCF, 32,000 sites were analyzed. So people will look at this output VCF and they'll say, well, there's 32,000 sites. Why are you saying only 14,000 are passing? And that's because most of these sites are where chimeric molecules happen to line up, or there were repeats in the genome, and we've decided they just aren't true deletions. And this is showing like the breakdown of the filters. And you can see that all of these filters are filtering out a fair amount of variance, and in combination, many of the filters are correlated with each other. So this sort of gives you an expectation. Only 43% of those sites were actually passing the filters in GoNL. So if you're observing something like this in your data, it's probably pretty typical. The same sort of metric happens when you go into genotyping. We put 14,000 sites into genotyping in GoNL, and 66% of those passed the genotyping filters. These are some of the reasons that they didn't pass. Some of them were just too short, or they were in repetitive sequence. We couldn't get a strong enough signal from read depth and various things. Some of the sites turned out to be duplicates by looking at the posterior likelihoods. So, and the majority of the sites that are not passing these filters, they're probably good deletions. They're just too short to get accurate genotypes in each sample at the depths of sequencing that was available. Another thing you can use in Genome STRiP, one of the annotators is called variance per sample. And it's just looking, especially looking at this post-genotyping is very useful. And you just see how many variants you called in each sample. And depending on your populations, you may see some stratification here. But if you see a sample like this that's a huge outlier, well, there was probably something wrong, right? There was a funky insert size distribution, or your read depth was highly variable across the genome in that sample. Something like that went wrong, and you should look hard at those calls and maybe filter this sample. You can also use other annotators in Genome STRiP to look at the allele frequency spectrum of your data. Sort of the neutral expectation is that this should probably be about linear. Genome STRiP is actually very good at calling rare variants. This was a trio design, so the singletons with only one allele are depressed because the child is going to not have singletons unless there's not Mendelian inheritance going on. But in general, Genome STRiP is not terribly sensitive to frequency. It will call singletons at about the rate you would expect. It's much more sensitive to length. The other thing you can do is you can compare to previously ascertained sets. There's a reciprocal overlap annotator that's very useful for comparing two VCF files. You can look at whether or not you're able to assemble breakpoints, or if you have heterozygous SNPs that are occurring in hemizygous deleted regions. And one final tool that's very nice for looking at site by site is there's a tool called Plot Genotyping Results, and it generates a plot that looks like this for a particular site. So this was a particular site, happens to be from 1,000 genomes, and post-genotyping, you can ask for one of these plots. And what this is showing you is a histogram of the normalized read depth signal. You can see that there's 3 nice clusters here. This is a pretty high frequency deletion. The samples in green here were confidently called as heterozygous deleted. The orange are confidently called as, I'm sorry, these are homozygous deleted. The orange are confidently called as heterozygous deleted. The gray samples, these are going to have genotype likelihoods, but they're going to be below 95% confidence because they were falling in between the copy 1 and copy 2 clusters here. And these little black things that may look like ants from the back of the room, these are actually little numbers telling how many split reads or aberrant read pairs are supporting each sample. And you can see that these are concentrated in the samples that are carrying the deletions. So this is additional evidence that this site was well genotyped, and you should be able to trust those genotypes. And even these samples in gray, you will find that the likelihoods are probably very well calibrated for these gray samples, so they will do very well in imputation. And this is a small site. This is only about 2 KB, and this was done in 4x data. The clusters and the genotyping would get better with increased depth. This is a larger site, about 8K, and you can see it's very rare. There's just one sample out here. It has definitely decreased read depth. It had 2 supporting read pairs. And we get 100% call rate at this site because we can be very confident about all the genotypes. So this is a very nice tool to go through site by site. If there are sites that failed filters and you want to take a look and see maybe why they failed, or if you have a particular site of interest, these plots are really useful. I'm going to talk just a little bit about some work we've done to support a very common usage scenario, which is to genotype a novel site, and in particular to genotype it in 1,000 genome space 1. So the issue that comes up is that maybe you've discovered a large deletion, maybe from array data or for some other reason you've got some samples and you found a large deletion. And so, especially maybe if you're looking at a Mendelian syndrome, you want to know how rare is this variant, right? I found it, but has anybody else seen it? You can go into the genome browser and you see a C, and maybe there's a couple DGV things there, but they don't quite look the same. So you might like to ask, was there any evidence for this site in 1,000 genomes? You could just look in the 1,000 genomes call set, but the drawback to that is if there was a false negative in 1000G, we just weren't confident enough to call that site, maybe you wouldn't find it there. So it would be very nice to have an inexpensive way to just ask, was there any evidence for this site, even though 1,000 genomes didn't call it? And if there was any evidence, how did it break down across populations? So we've done some work to make this kind of genotyping very inexpensive. So the 1,000 genomes data itself is publicly online, right? You can get it. It's on Amazon S3. It's hosted as a public data set. It's also available through HTTP or FTP from either NCBI or EBI, depending on which side of the pond you're on. And not much data is needed to genotype a single site, so accessing this data remotely is actually fairly practical if you're doing a small number of sites. And so we've added the capability of Genome STRiP that you can access remote BAM files over any of these protocols, and you can use those in genotyping. And then the other thing you need for genotyping is you need that metadata. But because we've already processed the 1,000 genomes, we simply packaged up the metadata from 1,000 genomes, so it's all pre-computed and it's on our FTP site. It's about 20 gigabytes, but that's not very much to download. You download this 20 gigabytes, and then in a few minutes, you can genotype a single site against this public data. And we've just very recently put on our website the beginning of a cookbook with little recipes and featuring this recipe for how you would go about genotyping a single site. So to genotype this site, what you would actually do is create a little tiny VCF to describe the site. You'd put in the chromosome and the start and end coordinates. I kind of made this up. This isn't a real site. I suppose you could try to genotype it. I suspect it's all ends in the reference. And then you run this through Genome STRiP genotyping against these remote BAM files. And what you get out is that Genotype VCF with genotypes from each of the 1,000 genome samples. And the cookbook recipe that's on the web actually also, if you just follow the script in that recipe, it will also generate the genotyping plot for you so you can go in and look at it. And this is an example plot from and there's also an example in that cookbook recipe where we genotype this site from 1,000 genomes. So you can genotype it and see if your plot looks like this, and then you kind of know that you've got things right. So a little bit just to wrap up, because I'm running out of time. This is where you can get the software. This is our website. We do require you to register, and the main motivation for this is that our funders really want us to know, really want to know from us, you know, is our user base increasing, and are people happy with the software? So we want people to register so we know that you know that you downloaded the software, and we encourage you to sign up and give us your email address. We have a support mailing list, and increasingly we've been using the GATK support forum and monitoring questions and answering questions on there. When you download a Genome STRiP, it's just a tar file and you untar it. It's pretty simple. There's also an installation test that comes. It takes about 10 minutes or so to run. It's just a toy data set, but if you have everything installed and configured right and your path is right, then the install test will run, and it will say it ran correctly. People also use this as a recipe for like designing their production analyses, and it's okay. I'd suggest that it's better to look at the examples that are in this presentation. And some of the common pitfalls when people try to adapt the install test is that it only runs single-threaded. It's not using the parallelization options that were in some of the other examples. It doesn't, to keep it at 10 minutes, it doesn't run reduce insert size distributions, but if you have any sizable data at all, you want to run this because all the rest of your processing will be faster. And it uses a -L1 flag, which if you know the GATK, you know this is saying only run chromosome 1. So people are surprised, they take the script, they copy it, and they say, how come I didn't get any calls on chromosome 2? Well, it's because you said -L1, but people don't know what -L1 means, they figure it's just some flag. So those are sort of common pitfalls. I've talked about the fact that Genome STRiP just is designed for whole genome processing, not for exomes. Its original purpose was to genotype many, many samples in populations. You can also, in theory, take a single deep coverage individual and call that in a background of many, many lower coverage reference samples, like 1,000 genomes. We're actually experimenting with this right now to try to give more concrete recipes and advice about how to do it. However, I can tell you that even in 1,000 genomes, the range of coverage in phase 1 ranged from about 1x to about 8x in certain releases. And Genome STRiP worked just fine with samples between 1 and 8x. We want to sort of check and see how it works with like 30x versus many 4x samples. But in general, it seems pretty robust to differences in coverage. So one way to call a deep individual is to round up 50 other individuals from 1,000 genomes, call your single deep sample against those 50, and then sort of ignore the genotypes from the other 50. In terms of resource requirements, this gives you sort of some ballpark examples of what it takes to run Genome STRiP. It's not cheap, but it is parallelizable through Q. So for example, if this was phase 1 of 1,000 genomes, 1,000 samples at 4x each, and the total here is probably, what is it, 600 CPU days. But if you run that in parallel, you know, on the BroadE cluster, this would probably run in about 6 days. And we are working on a bunch of scalability improvements. And in fact, I think it was around 6 to 7 elapsed days it took me to run phase 3 of 1,000 genomes, which is about 10,000x. So in summary, Genome STRiP was designed in the 1,000 Genomes project, and it's performed really well in all the benchmarks there and in all the validations. Other large projects have used it successfully, and an increasing number of projects are using it. We've had a lot of new users register in the last year. Probably the most common usage scenario so far, at least that I know of, is sort of, you know, doing sort of de novo discovery and genotyping on fairly large data sets. But you can also go in and genotype known deletions. Actually, what I want to talk about is you can do this efficient genotyping against public data sets. And we do this a lot. If there's some new suspected locus that comes up, we'll go in and we'll re-genotype it against 1,000 genomes and see if it looks like it was polymorphic there and just missed, or if something else is going on at that locus. Or if you can often like genotype a locus, and you can be rock solid sure that no sample in 1,000 genome carries that locus. And that gives you information that this probably is a pretty rare variant, right? Because across all the 1,000 genomes population, nobody has it. And we have a bunch of improvements ongoing that you can look for. And I wanted to thank Steve McCarroll in particular, and other collaborators here at the BroadE, the GSA and GATK team, because we ride on top of the GATK. The Genome of the Netherlands project let me share some of their data as an example, and there's a huge group of great collaborators in the 1000 Genome Structural Variation group. So that's all I have. Thanks very much, and if you have questions, let me know. Yeah, go ahead. Yeah, we looked a little bit at exome data in collaboration with Menachem, actually, when he was working on XHMM. And so Menachem's going to talk about this a lot more. There's a couple problems. One of the main problems is that the capture process itself introduces a lot of bias in read depth. So you'll have very different read depth across the different capture targets. There's a lot of different efficiency in the capture. So you have to have some way to normalize that read depth. And the other thing is that the Genome STRiP, because it's using a lot of read pair information, if the deletion breakpoints are actually in the intronic regions and they're outside your capture, all you have left to look at is depth. Now you might catch some deletions where the breakpoints happen to land in your capture targets, but that would be pretty unlikely. So in general, it's not a particularly efficient tool, and because you can't normalize that read depth terribly well, we just don't try to call on exome data. So a tool like XHMM is a lot better for exome data. That's a very good question. I actually get asked this fairly often. And the answer is, I sort of alluded to this a little bit. Genome STRiP, frequency doesn't matter too much in Genome STRiP. The main thing that determines whether or not you'll call a variant is how long it is. And then what length of variant you can call is directly correlated to how much depth you have, right? So in 1000 genomes, we do pretty well at 4x calling down to about 1 kb. Between 1 kb and 500 bases, we start to lose sensitivity. And you can see our sensitivity fall off. We'll call things down to like 300 bases in this 4x 100 base pair data. But our sensitivity is dropping way off by the time you get down to 200 or 300 bases. And it's pretty linear, right? If you double the coverage, you'll have the length scale at which you can call with the same power and the same efficiency, all other things being equal. So deeper coverage is going to give you smaller events. Yes, the deeper coverage, the large deletions just get better. When I showed you those two plots of the 2K versus 8K region, what you're seeing there in the tightness of those clusters is just having 4 times the data in the 8KB event. And if you imagine you take that 2KB event and shrink it down to 500 bases, you're only getting a quarter of the information in there. And so it gets much harder to accurately calibrate the genotypes. That's another actually very good question. I actually had thought, I forgot to do it at the last minute. I was going to put in a slide about our recent experience in phase 3 of the 1000 genomes. So in general, our belief was the more samples, the merrier. Although when I got to phase 3 of 1000 genomes, I called all 2,500 samples together on chromosome 20. And I saw that our sensitivity seemed to be dropping a little bit. We weren't recapturing some of the sites we had called in phase 1. And so I did an experiment where I called them in batches of 500 samples rather than all 2,500 together for discovery. For genotyping, I don't think it makes any difference, but for Genome STRiP discovery, and we got about 8% more sensitivity calling in batches of 500. And to be honest, I'm not quite sure why that was true. Actually, we've found, we've been looking into this. We know partly why it's true. There's actually an algorithmic limitation today for performance. If the read depth gets too high, we will punt a more expensive algorithm that tries to sort through areas of high depth and find signal even though there's a lot of noise in that area. And we punt today if the read depth gets too high. And our belief before was that a lot of those regions wouldn't have real deletions, but it turns out that a fair number of them do. So we're going to work on the scalability of that algorithm. So I think the cutoff there is, I don't know for sure at 500. So 500 4x samples was working pretty well. So that's about, what, 2,000x. So I might recommend, for very best results, calling in groups of about 1,000 to 2,000x, depending on your sequencing depth, and maybe calling with a minimum of, say if you call that 100 samples, 2,000x, that's 20x. 50 or 100 samples, or 1,000 or 2,000x, something in there I would aim for as a minimum. We do two things. We look at the mapping quality. There's a filter that the default is we look at reads that are mapping quality 10 and above. And then we also use the Alignability Mask. And there's quite a lot of overlap between those two. But we use the conjunction of those two. Yeah? Yeah. Genome STRiP will actually do a very good job on singleton deletion. So a single allele in 1,000 samples. What matters? Yeah. What matters more is the length, right? Like a 20 KB deletion. What matters is the length and also whether or not there's read pair evidence. So if there's a lot of unique sequence at the breakpoints, Genome STRiP will lose power. A read depth method will do, a pure read depth method will do better there. We're actually working on a pure read depth method, but it's not available yet. So maybe we should bring Menachem up because I think it's about time.